This conference will now be recorded. This conference will now be recorded.
ఇది పోయింది చూడు ఒకసారి ఇక్కడ నాకు
అయినా మేడం అని చెప్తాను.
recording progress already.
This conference will now be recorded.
Recording చేస్తున్నా. లేదు రెడ్ ఉంది అంటే Okay, today we'll start our class with inheritance and mutations. So learning objectives today will be the learner will be able to state the principles of heredity, explain the dominant and recessive and sex-linked inheritance with examples, and you should be able to classify the mutations and illustrate with examples. So what are the basic principles of heredity? So heredity is transmitted from the parents to the offspring as individual characters. Now the genes in the chromosomes, they will be linearly distributed at fixed positions that are known as the loci. Now genes that replace one another at the same locus, they are called as allelomorphic genes or alleles. Now alleles are genes that are responsible for the alternate or contrasting characters. Usually one allele will be inherited from the father and another one from the mother. So the meaning of this alleles is that for each gene you'll have at least 2 alleles. So one will be inherited from the father and one will be from the mother. And it does not it is not compulsory that both these alleles be exactly the same. Now genes on the chromosomes, they are linked and the linkages are more pronounced in the nearby genes. So and the character that you observe through this gene that is expressed by the gene is called as the phenotype. While the genotype represents the set pattern of genes present in the cell. Phenotype means what is the individual expressing, the particular character that is expressed by the particular individual, that is known as the phenotype. Now when both the alleles, if they carry the same defect, suppose if some defect is present in the allele and both the alleles that are inherited from the parents, both the parents, they carry some defect, then it is called as homozygous. But if one of the genes from one parent is normal and the other counterpart that is defective, it is called as heterozygous. So one allele, if it's normal, it's not exactly one gene. If one allele is normal and another allele is abnormal, then it is called as a heterozygous state, or you can say carrier state or the trait of the disease. So as I told you, for every gene you can have 2 of the same alleles. So either the alleles will be same or you can have different alleles. So when you say allele, either the allele may be recessive or it may be dominant allele. And if you see a recessive allele will only produce a phenotype if there is no dominant allele present. In other words, it means that only if you have 2 copies of the recessive allele, then only you can express the trait. So you need 2 copies of the recessive allele in order to have that trait that it codes for. So, and recessive alleles, they are symbolized by lowercase letters. Now, if you see in this table, you can see this is a pair of alleles for one parent, a pair of alleles from another parent. So when you will see what are the different types of combinations that you can get. So what we are saying is small a represents the recessive allele and bigger A represents the dominant allele. So both dominant alleles can become to one person. 2 persons can have one of each. Another person can have the recessive trait. So a dominant allele is one that is expressed to a greater degree than the other one, the other allele that is present. That's why it is said to be dominant. Now, when you talk about inheritance, as everybody knows that we have 22 autosomes and 22 pairs of autosomes and 2 pairs of sex chromosomes. So, which is the sex chromosomes may be XX or it may be XY depending on the whether it's a male or a female. Now, if you see, there will be now genes will be present on each of these chromosomes. So, a pair of genes will be present for a particular character in a pair of genes. So, one gene will be inherited from the one parent, another gene from another parent. So, here you can see this is a figure showing 2 homologous chromosomes. On one chromosome, there is a gene that is coding for red hair and another that is coding for brown hair. So now which person will get that particular person is having a gene for both red hair and brown hair. So whether a person will get red or brown hair depends on whether it's a dominant or it is recessive. Now if you see out of these 2 which is dominant, that will be expressed as a phenotype. And then if you see here, so here they have shown that brown is a dominant one, so brown hair is the phenotype. Now here both the parents, the gene is being expressed as a tall. So this will have the person will be tall only, the phenotype will be tall. So depending on whether it is dominant or recessive allele, the particular trait will be expressed. Now what is autosomal dominant inheritance? We will see now. Suppose if a father is an affected heterozygote. So you can see this gene capital D is represented as the abnormal allele and smaller d we are representing here as a normal allele. So this father is a heterozygote having one normal and one abnormal allele, while mother is having both normal. So mother is normal. So now what will be the children here? So if you see according to this table, what you have, what are the different types of combinations that you can get. So here you have 2 will be heterozygotes and 2 will be again be normal. So you can see, or you can see these 2 are the affected ones. So this the one they are receiving the trait because it's a dominant one, 2 offspring or 50% of the offspring will be affected, while 50% will be normal. An example of dominant inheritance you can see in some conditions like dwarfism or chondrodystrophy or some types of porphyrias. Then autosomal recessive inheritance. Here in autosomal recessive, here we give an example of where both the parents are carriers or heterozygous. So here N is being here the capital N. Now in this picture it is represented as a normal allele, while the smaller N is an abnormal allele. So that is, it is recessive. So now what is the combinations that you can get here is you can get here, this is NN, that is a normal. So one person is normal here, and you can see this is a capital and a small L. So that means 2 have become heterozygotes or carriers, while only one person individual will be affected. So this is the recessive trait. So they're having both the abnormal alleles, so they will be recessive. So the results of the offspring is that 50% will be carriers, 25% will be affected, and 25% will be normal. So this type of autosomal recessive inheritance you can see in conditions such as sickle cell anemia and most of the inborn errors of metabolism like phenylketonuria, or you can have albinism or galactosemia. Now we have said that sickle cell anemia, it is having an autosomal recessive inheritance. That means because both the alleles for the beta globin gene have been mutated, it means that all the beta globin genes will be abnormal. So this person will be homozygous for the sickle cell disease. But we also have a condition that is known as a sickle cell trait. So in certain cases, what will happen, the carrier state may be identified biochemically and it is referred to as the trait of the disease. So in this sickle cell trait, one beta globin allele will be normal while the other one will be abnormal. So such individuals are said to be heterozygous. So now what will happen, the normal gene will produce the normal hemoglobin and the abnormal gene will produce the hemoglobin S. So that means that in the RBC, 50% of the hemoglobins will be abnormal and 50% will perform the normal function. And this type of disease can be identified by means of electrophoresis. Then there is other than autosomal dominant and autosomal recessive inheritance, there is also another condition that is a type of inheritance that is known as sex-linked or X-linked recessive inheritance. Now in autosomal conditions, what happens? The disease will occur with equal frequency in both the sexes. So both the males as well as the females may be affected with equal frequency. But in sex-linked condition, what happens? The X chromosome will always carry the abnormal gene. So in X-linked recessive condition, the normal X will dominate over the abnormal X. That means if both the X's are present, then the normal one will dominate over the abnormal X. But in case of XY, what will happen? The abnormal X is expressed when present with Y. So here we'll receive in an example, if a marriage has taken place between a male who's having a normal gametes and a carrier female, what are the different possibilities that may happen? So this is the female XX and this is the XY male gametes. So what will happen? You may have XX here and another XX here because this is abnormal abnormal gene that is present in the X. So one will be a carrier and another daughter will be normal. While out of the two sons, one son will be affected because of the abnormal gene and another one will be normal. So what is happening again here? The probabilities are 1/4 of the male will have one disease, 1/4 of the female will be a carrier, 1/4 will be a normal male, and 1/4 will be a normal female. Then if you see a person, if suppose the male is having affected gametes and the female is having the normal gametes, then the possibilities you can see here is that, so from the affected male, both the daughters are getting the abnormal this one gene, but because of the normal X, this will dominate over the abnormal X. So that is why they are carriers. So both the daughters will be carriers, while the sons, what will happen? Because the sons are inheriting the normal X, they will be normal. Thus both the sons will be normal. Then suppose if the male is affected and the female is a carrier, then the possibilities are one daughter will be affected, one daughter will be a carrier, and one son will be affected, and then one son will be normal. So that means one daughter and son will be affected, one daughter will be a carrier, and the son will be normal. So the examples of X-linked recessive inheritance we can see in diseases like hemophilia, G6PD deficiency, or Duchenne type of muscular dystrophy, or red green color blindness. Now there is also variable phenotypic expression can also be seen, because all the genetic conditions, they are not congenital. That means they are not present at birth. People may also acquire these conditions during the adulthood or during the childhood. So even dominant characters may not show 100% penetrance in actual practice, so that an affected parent, it is not compulsory that affected parent will have an affected child. So affected parent may not have an affected child, or the offspring may just carry the mutant gene. That means they are just the carriers, and but they'll be phenotypically, there will be normal. Now, when the phenotypic expression of a mutant trait occurs only in the adult life, it is called as the age-related penetrance. So, example you can give of triplet expansion defects that is seen only in the adulthood. So, in dominant condition, variable expression of critical features may be seen in different family members. Now, the location of many genes have been identified on specific chromosomes. So, genetic difference, because of genetic disorder, therefore, they are of different types, depending on the way the abnormality on the gene is. So, there may be chromosomal disorders, which have been identified by karyotyping. Example, you can give of 21 trisomy. Then you have single gene defect. Sometimes that can be identified also by biochemical methods. Example is phenylketonuria, where the gene encoding for phenylalanine will be abnormal. Then you may have mitochondrial abnormalities or several multifactorial disorders. So all these are the different type of genetic disorders that can be seen. Now, several genes have been identified in the human chromosomes. So you can see here, depending on chromosome 1, in chromosome 1 you have the gene for alkaline phosphatase. Then if you see in chromosome 4, you have the genes for alcohol dehydrogenase fibrinogen. Then if you see for in chromosome number 18, you have the gene for prealbumin. So several genes have been identified coding for particular proteins. And you can see here in X chromosome, you have glucose-6-phosphate dehydrogenase, anti-hemophilic globulin gene, HGPRTase gene, so and so on. Now, there is already we have spoken about chromosomal recombination several times. Here also exchange of genetic information that can occur between chromosomes can occur. Now, if it is equivalent crossing over takes place, then it's normal. But sometimes unequal crossing over takes place that may result in disorders may be seen. So unequal recombination will produce some condition like lipohemoglobin, which is an abnormal hemoglobin. Then we will see what are the consequences of DNA damage. So we have talked about what is the type of inheritance that you may get, and what are the abnormalities, what are the abnormal different types of inheritance that may occur because of the abnormalities. So there may be dominant inheritance, there may be recessive, or maybe excellent inheritance may be seen, or there may be some variations. So what are the consequences of DNA damage? So DNA damage, it will lead to mutations. And then what do you mean by mutations? So there is a mutation means there is an alteration in the genetic material. So we can define mutation as a change in the nucleotide sequence of the DNA. This change in the nucleotide sequence, either it may be cross, so that large areas of the chromosome are changed, or they may be very subtle, with a change only a one or a few nucleotides. Then mutation you can also define as an abrupt, spontaneous origin of new character. Mutation in the germline is transmitted to the offspring. Now, one thing you have to remember that mutation is always not that is genetically transmitted. Sometimes mutation may occur when an individual genome, there is an interaction between the individual genome and an environment you are exposed to. So because of that also mutations may occur. So that's why we're saying it may be abrupt spontaneous origin of new character, or it may be genetically transmitted. Statistically, it has been said that out of every 10 to the power of 6, this is sorry, not 1, not 6, 10 to the power of 6 cell division, one mutation takes place. That means 1 in every million cell divisions, one mutation may be seen. Now, a mismatching of the base pairs can occur at a rate of 1 per 10,000 base pairs. Then DNA polymerase is always there to proofread and repair accidental mismatch repairs. And chances of a mutation occurring at any one gene is 1 in 100,000. Now, because of the human genome being so large, even at this stage, mutations do add up. So, probably each one of us have 3 or 4 mutations. Now, what are the effects of mutation? So, the mutation will cause a change in the DNA. So, change in DNA will affect the genetic information. Then they can also cause change in the, because of the change in genetic information, they can cause a change in the enzyme activity, they may change the nutritional requirements, antibiotic susceptibility may be changed because of that, morphology, antigenicity, and many other properties of the cell will be affected. So the mutation, they can lead to genetic diseases. Then this what are the causes of this mutation? So the what are the causes that lead to mutation? They are known as the mutagens. So mutagens may be chemical or physical agents that interact with DNA to cause mutations. So physical agents may be high energy radiation like X-rays and ultraviolet rays, or they may be chemical mutagens, which falls into several categories. Now, these chemical mutagens may be base analogs. They may be base analogs that may be substituted for the normal bases in the DNA, and they will pair incorrectly during the DNA replication. Or they may be interference with the DNA replication by inserting into the DNA and by insertion of these chemicals into the DNA and distorting the double helix. Or the bases itself, the changes, chemical changes may occur in the bases itself. So that this will again lead to changing in their pairing properties. So other than the physical and chemical agents that are mutagens, you may also have viral mutagens. This also we have seen, and when we are talking about cancer, where a number of tumor viruses were identified in animals as well as in humans. And about 15% of the human cancer are caused by the viral infections that disrupt the normal control of the cell division. And we have already discussed before how the tumor viruses transform the cells into cancer cells through the integration of the viral nucleic acid into the host cell DNA. Then what are the types of mutation? So a point mutation, it is defined as a change in a single nucleotide. And this change in the single nucleotide may be subclassified as substitution, deletion, and insertion. So all of these changes may lead to either missense, nonsense, or frameship effects. So first we'll see what is a substitution. This is a very common type of mutation where a single base alteration will be seen, and or a substitution of a single base may occur. This substitution of a single base is of two types. It may be transition, where a given pyrimidine that is present in a codon may be changed to another pyrimidine, or a given purine is changed to another purine. So pyrimidine is changed to a pyrimidine only, or a purine to another purine. While in case of transversion, the changes that take place are a purine may be changed to either of the two pyrimidines, or a pyrimidine may be changed to either of the two purines. So here we are showing that what is having these are the pyrimidines, where thymine may be replaced with cytosine or cytosine with thymine, and the purines adenine by guanine, it may be replaced or guanine by adenine. So when a purine is replaced by a purine or a pyrimidine with pyrimidine, then you call it as a transition. Or in case of transmission, here you can see the thymine may be replaced by either of the two purines, that is adenine or guanine, and cytosine similarly by adenism guanine. And here you can see these two purines, adenine and guanine, will be replaced either by the pyrimidines, thymine or cytosine. So these are the transversion. So here in the example, you can see here adenine has been replaced with cytosine. So this is an example of, so here purine is being replaced by pyrimidine. So this is an example of transversion. Then what happens? What are the consequences that may occur as a result of this substitutions? So it may result in silent mutation because the codon that containing the change base may code for the same amino acid. So example you can take here, UCA codes for serine. Now if the third codon or third base is in the codon is changed from A to U, this UCA also still codes for serine. So that means now this mutation is not going to be, it's not going to affect the function of protein. That's why we call it as a silent mutation. Missense mutation is where the change base may code for a different amino acid. So if you take the same example, UCA that codes for serine. Now here the first base may be U may be replaced with A. So if it is substituted for A, what will happen is that now this ACA will code for threonine. So it is a different amino acid. Now this missense mutation, it may be acceptable, it may be partially acceptable, or it may be unacceptable because it is concerned with regard to the function of a protein. Then nonsense mutations are those where the codon will containing the altered base may become a termination or a nonsense codon. So example, the second base, so still continuing with the same example, serine UCA. So now the second base, if it's changed to A, then this UAA becomes a stop signal. Now you can see here with an example here, this is a codon here, that mRNA that is coding for AUG codes for methionine, AAG for lysine, UUU for phenylalanine, and GGC for glycine. And this is the stop codon. Now starting here with glycine, we'll see the third base C, if it's replaced with U. So GGU still codes for glycine. So this is a silent mutation. So the function of the protein is not going to get affected. Now if you go to the second one, here the same phenylalanine for glycine, the first base G, if it's changed to A, this AGU codes for serine. Now this is another amino acid has come instead of glycine. So this again I told you missense mutation may be acceptable, partially acceptable, or it may be unacceptable depending on the function of a protein. Then nonsense mutation here. So here we are taking here example of lysine here where the A is replaced with U, it becomes a UAG becomes a stop codon. So that means the protein is terminated here itself. It's terminated prematurely. So these are other examples of the silent mutation, missense, and the nonsense mutation. Now coming to what are the diseases that may arise as a result of this substitution? Again, here we are taking an example of sickle cell anemia here, where you can see the codon GAG, the A is being substituted with T. So the glutamic acid, GAG codes for glutamic acid, it is being replaced by valine. So this GUG is coding for valine. So the glutamic acid is replaced by valine at there is a change in a single amino acid because of a change in a single base. So what happens is that change of a single nucleotide in the DNA's template strand leads to production of an abnormal protein. So here we have given an example of hemoglobin, where what are the effects of a single base change in this protein. So if you see hemoglobin, this is adult hemoglobin, the beta change. If the 61 at the position 61, if lysine is replaced with aspargine, it results in an abnormal hemoglobin that is known as hemoglobin Hikari. Then again here the partially acceptable, or this is missense mutation that is used, seen this is an example of sickle cell anemia, where in the beta chain, at the position 6, the glutamate is being replaced by valine to form hemoglobin S. That is sickle cell hemoglobin. Then you have a third unacceptable missense mutation. So this is acceptable hemoglobin Hikari, where that means the function of the protein will be almost normal. Then partially acceptable, it is still partially, it can still function. Unacceptable is here where in the alpha chain at position 58 histidine is being replaced by tyrosine. So what is happening is that the abnormal hemoglobin that is resulting is known as hemoglobin M. So this is here you can see hemoglobin N in methemoglobinemia, which is an abnormal protein and cannot function. So this is the same example of what we have seen here, unaccepted missense mutation where histidine is being replaced by tyrosine. So a tyrosine may be mutated to a termination codon. So it is being terminated to termination codon UAA or UAG. So here UAA or UAG it may be coding for. So this leads to premature termination of the protein, and so functional activity will be lost. Example you can see in beta thalassemia. Or a terminator codon is altered into a coding codon. This will result in elongation of the protein to produce run-on polypeptide like hemoglobin constant spring. So here actually this is not an example of what I told you about beta thalassemia. Here I'm just showing that tyrosine again, it is being replaced. It may be mutated to a termination codon in case of beta thalassemia. Now this insertions and deletions, if we see, we talked about substitution. So we will talk about what are the insertions and what are the deletions. So inserting or deleting one or more nucleotide will result in frame shift mutation. So the deletion of a single nucleotide from a coding strand of a gene will result in altered reading frame in the messenger RNA. So what will happen if there is a deletion of a single nucleotide? The machinery that is translating the messenger RNA will not recognize that a base is missing because there is no punctuation in the reading frame of the codons. Because of this, a major alteration in the sequence of the polymerized amino acid occurs. So this will result in a garbled translation of the mRNA distal to the single nucleotide deletion. That means there is no meaning in the translation of the messenger RNA, and this will happen distal to the single nucleotide deletion. Now, insertion of one or two or multiples of even 3 nucleotides into a gene will also result in an mRNA where the reading frame is distorted upon translation, and the same effects that occur with deletions will also be reflected in the mRNA translation. Now, reading of the message by either insertion or deletion can also result in the appearance of a nonsense codon. So even if you insert or delete, it may also result in the reading. When the reading frame reads, it may read it as a stop codon. So it results in the production of a polypeptide, both garbled and prematurely terminated. So here we will see an example. Here you can see that adenine, here CTGAGT is there. So when insertion is being done, after this adenine, a T base is being inserted, extra base is being inserted. Now if you see with deletion, this A base is being deleted from the sequence. So this is a deletion of a base. Now what will happen if insertion or deletion occurs? So we said frame shift mutation, alteration of the reading of the frame will occur. Now if you see this, previously it was ATG coding for methionine, GAA for glutamine, GCA for alanine, and CGT for glycine. Suppose if this G is deleted, So if G is deleted, what will happen? The reading frame changes. So now this AAA will become AAG. So this reading frame will bring this one base into this reading frame. So it will become AAG, and this will become CAC, and this will be GT. If this is the last sequence, that means one base is less over here. So this, instead of glutamic acid, you will have lysine. Instead of alanine, you have histidine. So that means the amino acids will be changed in this frameshift mutation, and the protein synthesis will have several altered amino acids, or they may be prematurely terminated proteins. So here you can see, in case of frameshift mutations, here we have an example of again methionine AUG, lysine AAG, phenylalanine UUU, and glycine CGGC, and this is a stop codon. So here, if you see, if there is an insertion, an extra U is inserted in the before the lysine, after the methionine, what will happen? This will alter the reading frame of this one. So now this frame shift causes an immediate nonsense, that is UAA, that is with UAA is a stop codon. So it is causing a premature termination of the signal. Then if you say here, the U that is present here, in the phenylalanine, the U that is present here, it is deleted. Here we talked about insertion. Now if it's a U is deleted here, then what will happen? The next base pair will come over here. Instead of phenylalanine, you have leucine, and then it becomes alanine. So this is an example of extensive missense mutation in frame shift causing extensive missense mutation. Then insertion or deletion of 3 nucleotides may also occur. So when insertion or deletion of 3 nucleotides occurs, if insertion of 3 nucleotides means an entire one amino acid is being added, or entirely one amino acid is being removed, missing. So here we have an example where one entire AAG that was coding for lysine, when this 3 nucleotides may be removed, they may go missing. So you have only phenylalanine, after that methionine, after that phenylalanine, the lysine goes missing. So insertion or additions or expansions, they may be subclassified into a single base addition leading to frame shift effect, which we have seen now, or if there is a 3 nucleotides are being added, that is known as trinucleotide expansions. And this you may be seen in some conditions like Huntington's chorea, where this 3 base pair codon CAG trinucleotides, they are being adding continuously, so 30 to 300 times. So this CAG codes for glutamine. Now this leads to a polyglutamine repeat in the protein, and the severity of the disease is increased as the number of repeats are more. So instead of 30, it is 300. The severity of the disease will be more in case of Huntington's chorea. Then the base pairs may be duplicated. In Duchenne muscular dystrophy or DMD gene, duplication is seen in the disease. So this trinucleotide repeat expansion, what I gave you an example of Huntington's chorea. Other than that, you have also other disorders that are known as Fragile X syndrome or myotonic dystrophy. And they differ from the normal counterparts only in the number of tandem copies of a trinucleotides. So the expansion of this trinucleotide repeat in the mutant allele can be in a coding region, or it may be in an untranslated region of the gene, or even in an intron. So depending on where these repeats are there, it leads to different types of disorders. Then deletion may be subclassified into a deletion again of a single base, or which may give rise to again frame shift effect, or large gene deletions may occur, such as in the case of alpha thalassemia, where an entire gene may be deleted, or in case of hemophilia, where only a partial gene will be deleted. Or deletion of a codon may occur. Example in case of cystic fibrosis, where one amino acid phenylalanine will be missing in the CFTR protein. Now this large segment deletions, again we come back to the crossover recombination, where large segments of the DNA can be deleted from a chromosome during unequal crossover and meiosis. In a normal crossover, what happens? The homologous material and paternal chromosomes exchange equivalent segments, so that no genetic information will be lost from either of the chromosomes. But on rare occasions, unequal crossover may occur, so that one of the two homologs loses its genetic information. So, example I've already told you about alpha thalassemia, which may occur as a result of unequal crossover. Another condition is called as Cry-Do-Chat, which have characteristic presentation like with mental retardation, microcephaly, white set eyes, and a characteristic kitten-like cry may be seen. And this results from a terminal deletion of the short arm of chromosome 5. Then mutation may also occur in the splicing sites. So where normally what will happen, splicing will be seen during the post-transcriptional processing. And if a splice site is lost to mutation, the spliceosome, what it may do, it may delete nucleotides from the adjacent exon, or it may leave the nucleotides of the intron in the processed mRNA, or it may use the next normal upstream or downstream splice site, deleting an exon from the processed mRNA. So mutation in the splice sites leads to some disorders, that is beta thalassemia, Gaucher's disease, and Tay-Sachs disease, which are lipid storage disorders. So here you can see, this is where nucleotides from the introns may be less amount of new intron may be excised, or they may be include exons from the another exon may be deleted. They may be removed, excess bases may be removed, exons, so removing the exon nucleotide, or an exon itself may be skipped. Now this type of mutations may be lethal, where the alteration may be incompatible to the life of the cell or the organism. So this we can see in case of alpha-4 hemoglobin, which is lethal, and so the embryo will die. Or the mutation may be beneficial. So spontaneous mutation always occur during the evolution process, and such beneficial mutants are artificially selected in agriculture. So where gamma irradiation of the seeds vary to get more better variety of crops. Then carcinogenic mutation, where the mutation may not be lethal, but it may lead to alter the regulatory controls, leading to cancer. Now this also we have seen the how to test for mutagenicity is through the AMIS test, because here they use a salmonella strain that where the histidine is mutated. In this strain, the histidine will be mutated. So in order to grow, it requires histidine. So it is grown in a medium containing histidine, and mutagens are also, chemical mutagens are also added to the mix. And this strain will be very sensitive to this chemical mutagens. So the number of colonies that are grown, it will show the number of mutations. The more number of colonies that are grown, it shows that more number of mutations are there. So